Welcome back to the lectures for MLT 110. In this lecture, we'll be covering routine venipunctures. This lecture will be divided into two parts. This lecture will cover topics like the different collection tubes used in phlebotomy, the anticoagulants contained within those collection tubes, the order of draw. This is the order in which we must draw our different types of tubes during phlebotomy. The minimum volume requirements of each of the different types of collection tubes, and then just the overall venipuncture procedure. Okay, let's start off by talking about the different types of collection tubes that you can use within phlebotomy. The first tube that we want to talk about is the plain red top tube. This can also be referred to sometimes as the serum separator tube or the SS tube, or it also can come sometimes in a gold color or a tiger top color. This is a tube that is used for chemistry test usually. The blood, because there's not an additive in the tube, the blood is allowed to clot. The liquid portion after the blood has been spun down in the centrifuge is known as serum. If there ever is an additive in the red top tube or the plain tube, it is a clot activator usually, because again, we want this blood to clot in order for there to be serum in the tube. Here is an example of the plain red top tube, but again, sometimes it can come in other colors such as gold or the tiger top. The tiger top is a marbled looking color, and it's kind of a cross between a orangish red color and a gray. So that is the tiger top. Next on our list for collection tubes is our lavender collection tube or our EDTA tube. This tube is used for our hematology test usually, and hematology tests would include our CBC or complete blood cell count or our ESR test. And the additive is EDTA, and this is a chelating agent which binds with calcium, and this prevents clotting. So this does the exact opposite as our serum tube. It prevents clotting, where our serum separator or our plain top tube, it allows the blood to clot. The last tube on this slide is our blue top tube. This is the sodium citrate tube, and this additive also binds with calcium to prevent clotting. So you're going to end up with plasma as the liquid portion of the blood after you centrifuge. This tube must be filled to a certain level. It needs to have a 9-to-1 ratio of blood to anticoagulant in order for testing to be accurate. This tube is usually used for coagulation testing. Again, coagulation testing is testing to see how well the blood clots. And some of the tests that we do for coagulation are our PT, PTT, and D-dimer test. Again, here are our examples of our lavender top tube, which has the EDTA additive in it, and the blue top tube, which has the sodium citrate additive in it. These are both anticoagulant tubes that prevents clotting of the blood. And remember, this sodium citrate tube has to be filled to a certain level with the blood in order to have the right ratio of blood to anticoagulant, and there's usually some kind of mark or indication up towards the top of the tube that lets you know you have enough blood in the tube. The next type of tubes are our green top tubes. These tubes are usually used for chemistry testing. The green top tube can either have lithium heparin in it or sodium heparin in it as an additive. These additives inhibit thrombin to prevent clotting. So these are an anticoagulant tube. So after spinning your sample down, the liquid portion of the blood is going to be plasma. The lithium heparin can be used for general chemistry panels for a patient, but if a lithium test is ordered for a patient, you cannot use a lithium heparin tube to perform that test because the lithium in the tube will throw that result off. If a lithium test is ordered in clinical chemistry, you could use a sodium heparin tube or you could use a plain red top tube because it wouldn't have that additive of the lithium to throw the test result off for the patient. For the sodium heparin, it is also used in chemistry testing, but again, you couldn't use the sodium heparin tube if sodium was a test that was ordered, because the sodium in the sodium heparin tube would throw off the test or the result for the sodium, just like in the case of the lithium. So as a lab tech, you just have to be careful and know what additives are in what tubes so some of your additives don't interfere with testing results. Next is the gray top tube. It has potassium oxalate in it as an anticoagulant. It also binds with calcium. It also has a preservative of sodium fluoride in it. It's usually used as a tube for preserving specimens and for specimen transport. Some of the tests that we usually use the gray top tube for are the glucose test, the alcohol test, and the lactic acid test. Because this is an anticoagulant tube, the liquid portion of the blood after you centrifuge, you're going to end up with plasma. Now let's discuss order of draw. This is when you have multiple tubes be drawn at one time, and it's recommended to draw your blood tubes in a certain order to prevent cross-contamination. The procedure is the same for all tubes and instruments used, and it's vital in order to not allow for cross-contamination and to ensure the accuracy of test results. The smallest error could mean that the patient might not receive the appropriate care and treatment. Okay, so what exactly is the correct order of draw? What do we need to draw first versus what do we need to draw last? If we have orders for blood culture bottles, we're going to draw those first. And you usually draw two blood culture bottles, an aerobic and an anaerobic. Next would be your coagulation tubes. These would be your blue top tubes. Remember, your blue top tubes need to be filled up to a certain point in order to reach the correct ratio of blood versus anticoagulant. Remember, it's a 9-to-1 ratio. And you can see here, you need to fill up to this line. In this particular image, this blue top tube is not filled up enough. You only have blood up to here, and then there's some bubbles, and the actual line is more in this area. So this tube would actually be rejected by the lab because it's not full enough. It doesn't have the correct anticoagulant to blood ratio. Next in the correct order of draw would be your serum tubes. These would be your plain red or your gold top tubes. These would be your tubes that could contain a clot activator, or they may be plain and contain no clot activator. So these would allow your blood to clot. You would actually need to allow these tubes time to clot before you spin them down, because you're going to want serum as your liquid portion of the blood after you spin it down in the centrifuge. Also, something to note, in your blood tubes, you can have an additive of a gel separator. And your gel separator can come in any of your tubes, whether it's a serum tube or a plasma tube. And this gel separator is just something that can be a dividing line after you centrifuge your tube. It can separate your serum or your plasma that's on the top from your red blood cells that's on the bottom of your tube. And again, it's just a little layer of gel that's separating the liquid portion of your blood that's at the top, being whether it's your serum or plasma, depending on what type of tube you have, and the red cells that's been spun down to the bottom of your tube in the centrifuge. The heparin tubes are next in your order of draw. These are your green top tubes. These are your tubes that are used for chemistry testing. Next would be your lavender top tubes. These are your EDTA tubes. They're usually used for hematology testing. And finally would be your gray top tubes. They are the last tubes in your order of draw. Let's talk about blood culture bottles a little bit more in depth, because this would be the very first thing that you would draw in your order of draw if there's orders put in by the physician. It's usually recommended to draw a waste tube before ever drawing your blood culture bottles to prevent contamination. A lot of times we use a tube known as an SPS tube. This is a yellow top tube. This tube can be used to empty into the blood culture bottles if needed. But if blood is emptied from the SPS tube into the blood culture bottles, the blood should have only been held in that SPS tube for only up to 4 hours. Some things to note about the SPS additive is that it's shown to be an anticoagulant, and it can be an inhibitor of some cellular elements, and it can interfere with some bacterial growth. So it might be better just to go ahead and collect your blood cultures directly into your blood culture bottles. When we're collecting blood for blood culture bottles, we have to use some special techniques. We have to use an antiseptic technique and do a little bit more in-depth cleaning than we would on a normal venipuncture collection. Instead of just cleaning with alcohol, we're going to clean with either iodine or chlorhexidine to prevent contamination from the skin. And we're going to clean the site for at least 1 minute. The reason we're so adamant about cleaning and making sure the surface of the skin is clean is because we want to truly know if there is bacteria in the blood. We don't want any bacteria contaminating our sample from the skin surface. Again, we're going to collect 2 different blood culture bottles. One of them's going to be aerobic and one of them's going to be anaerobic. And each bottle is going to be collected from a different site on the body. So let's say we collect one bottle from the left arm, then we're going to have to collect the second bottle from the right arm or maybe the hand. It has to be a completely separate site. And when we're collecting blood culture bottles, we need to make sure we have enough volume or sample of blood in our blood culture bottles. There's usually a line that tells us that we need to reach as far as volume for our blood sample. And usually most bottles will require about 10 milliliters of blood in order for that bottle to be considered full. This is a really nice chart showing the different types or colors of the different tube tops that are available, and it also shows the different sample types that you'll end up with based on the tube type, whether you'll end up with serum, plasma, or whole blood. Also, it tells you the different additives that are in the tube and most common testing that's done with that type of tube. You might want to pause the video at this time and study this chart a little bit closer. So different blood tube types require different volumes of blood in order to perform a test correctly. Some are a little bit more sensitive than others. The red top, or the SST, the serum separated tube, is really not affected by the amount of blood that you have in the tube, as long as you have enough volume to complete the test or test you're wanting to run. The lavender top tube, or the EDTA tube, if you're using a smaller tube of 3 milliliters, you should at least have 2 milliliters of blood volume in that tube. In the larger 7 milliliter tube, you should have a minimum of 5 milliliters of blood in that tube. The blue top tube, or the sodium citrate tube, is the most sensitive when it comes to the volume of sample. It must be a full draw. Remember, you need that 9-to-1 ratio of blood to anticoagulant. If the blue top tube comes to the lab and it's not filled up to the fill line, unfortunately the lab is going to have to reject that tube, and that tube is going to have to be redrawn. Our last two tubes that we need to talk about are our green top tubes and our gray top tubes, and they're not really affected by the amount or volume of blood that are collected in our tubes, as long as we have enough sample to complete testing. This completes part 1 of our venipuncture lecture. I will see you over in part 2.